The green button on top is the clicker? Yes. Okay, thank you. So, yeah? 1 second. I'm Suzanne Jackowski, and I'm very excited to talk to you today about our... Oh, close to my mouth, okay. I'm Suzanne Jackowski, and I'm very excited to talk to you about the progress in our research program that's led to BBP 671. This is an investigational small molecule that is currently being studied as a potential treatment for PKAN. As for my disclosures, I'm a research scientist, and I recently retired from the faculty of St. Jude Children's Research Hospital in Memphis, Tennessee, where I worked for more than 40 years. For the last several years, I was the leader of a team of scientists who invented and developed the preclinical laboratory program for the series of compounds that led to BBP 671. This research was funded not only by St. Jude, but also supported by COA Therapeutics. I'm a member of the Scientific Advisory Board of COA Therapeutics. I'm a former member of the Scientific Board of the NBIA Disorders Association, and also a past member of the Tennessee Rare Disease Advisory Council, or REDAC. Now, PKAN is caused by mutations in the PKAN2 gene, and these mutations cause molecular changes, discrete molecular changes throughout the length of the protein that reduce, or in the most severe cases, delete the activity of the PKAN2. This is an important enzyme. And there's also happens to be, there are a wide range of symptoms among PKAN individuals, and this reflects in part the degree of inhibition or loss of PKAN2 activity. Now, PKAN2 mediates the incorporation of vitamin B5 into coenzyme A, and when you lose the activity there, we're in trouble. But PKAN2 has two backup proteins in all cells, PKAN1 and PKAN3. And normally, all three proteins work together to control the amount of vitamin B5, or panthenate, that gets converted into CoA. So these activities, the PKAs, can compensate for one another. And the activities are dynamic, and this allows them to change the CoA level as needed in response to molecular demand, which happens all the time, whether you're eating or fasting is one example. So I'd like to give you a little bit of a holistic view about coenzyme A. It was discovered in the 40s, and in 1953, Fritz Lipman was awarded the Nobel Prize for the discovery of CoA as an essential helper in metabolic reactions. In 1964, Bloch and Lennon won the Nobel Prize for linking CoA to lipid metabolism. And in the years following, several more prizes were awarded linking CoA or a CoA derivative, either directly or indirectly, to important essential processes. So we know that CoA is involved in hundreds of metabolic reactions throughout the body. But actually, different tissues, cell types, and organs require different amounts of CoA, depending upon what their vital function actually is. What is their readout? The readout from liver is very different from the readout from brain. The function of brain is very different from the function of the muscle. And the different organs actually prioritize those CoA-dependent reactions that are necessary for their particular vital functions. So So let's talk about neurons. PKAN is a neurometabolic disease, and research from our lab has shown that in animals, when there's a CoA deficiency in brain, there's a change in the brain metabolism. Now, PANK2 is an abundant PANK, particularly in brain and spinal cord. It's unique in this respect. And PANK2 is also unique because it associates with mitochondria. This is the energy powerhouse of the cell, and it's shown by the green dots on this microscopic view of the cell. The nucleus is designated in pink. Now, we know from a variety of studies that loss of PANK activity can lead to a CoA deficiency. Now, this deficiency can be temporary or transient because the other PANKs can compensate to some extent. But a consistent loss or a consistent deficiency in CoA can actually disrupt a vital function. And in neurons, we know from a variety of studies from different perspectives that a CoA deficiency causes problems in iron handling and iron homeostasis, nerve transmission, nerve development, and movement. So our laboratory strategy was to find an activator of one of the alternate PANKs. We chose to target PANK3 because it's expressed everywhere in all cell types as best as we can determine. And we had to develop techniques to measure CoA in cultured cells following addition of the compounds. After that, we went on and moved into animals to determine the pharmacological properties of several of the promising compounds. Now, this was a real scientific challenge because we had to design new methods and learn new technologies along the way. We had to solve this 3D structure of the PANK3 protein, not only in a static state, but as the protein moves through the chemical reaction, which it does. We were looking for an activator of PANK3, and most drugs on the market are actually inhibitors of the enzymes and their targets. We wanted to test all of the PANK proteins, and this required purification of a large amount of protein in each case. And we wanted a compound that would survive, that could be administered orally and could survive the digestive action of the GI tract and cross the blood-brain barrier. So this is just an illustration of the process. It was a real team effort, and we called in the expertise of several departments at St. Jude, particularly chemical biology and structural biology. And we designed an assay and did a high-throughput screen of all the chemical libraries that were available at that time, and that was more than 500,000 compounds. We took the hits and we filtered them computationally based on a novel approach that favored blood-brain barrier penetration based on experiences with past drugs. We settled on a scaffold, and we made we had hundreds of chemical variants of that scaffold, trying to maintain those principles that would favor blood-brain barrier penetration. And we tested their ability to bind tightly to the PANK3 protein using at least 4 different methods. And I've shown here just a cameo of the structure of BBP 671, which is the stick and ball structure bound to the pocket of PKAN3 protein. The compounds that looked good following our assays in the test tube, we then moved to cultured cells where we added the compound to the medium and determined whether it would bind PKAN3 in the biological context and raise the cellular CoA levels. Moving on from there, we went on to do the pharmacology in animals to determine how well the compound distributed through the body and whether it raised the CoA levels in brain. So BB671 is a newly developed small molecule, and we actually published our progress in several papers along the way as we worked on developing the best compound we possibly could. For example, PZ2891 that Dr. Mamoon referred to had very high target affinity. It bound to PKAN3 very tightly at the picomolar level. But it wasn't that great when it was in circulation. It actually cleared the system fairly rapidly, and it wasn't very stable. So we felt that this was not acceptable moving forward. So coming to PZ3022, we solved the problem. We went back to the drawing board, made more molecules, and tweaked the structure as best we could as guided by the structure of the PKAN3 protein. And we solved the problem of the metabolic stability in circulation, but in the process we lost some of the potency, and it didn't raise the CoA level as well as PZ2891 at the same dose. So we went back to the drawing board again, and we arrived at BBP671, which binds tightly to the PKAN3 protein. It has very high potency at the picomolar level, and it also has a very low rate of clearance in the circulation. And this allows the small molecule to stay around long enough to pass into the cerebral spinal fluid and go into the brain, and in sufficient quantities to do that. Now, with the help of CoA Therapeutics, this is some data confirming that orally administered BBP671 penetrates the blood-brain barrier and raises the brain CoA level in rats. In the graph on the left, you can see that within hours after the oral dose, the plasma levels of the BBP compound increase very nicely, and then they eventually, with time, disappear. But they stay around for a relatively long time. They're still above normal by 8 hours. And we also see a significant amount of the drug appearing in the cerebral spinal fluid, and this is at therapeutic doses. We also see in the graph on the right a nice increase in the small molecule appearing in the brain, and following that we see a substantial increase in the CoA level in the brain. Now there's a lag in the increase in the CoA level because following the activity of the PKA, there are 3 more enzymatic steps that are required to make the final coenzyme A molecule. As another precursor to our anticipated phase 1 study in humans, we figured out how a type of biomarker in the blood that we could monitor to evaluate the response of the following oral administration of BBP671. We did this because we cannot regularly do tissue biopsies to measure CoA levels in human. So in the graph, here are the acetyl-CoA levels in blood in control, which is before administration of the compound, and at 4 hours after dosing with increasing doses of BBP 671. And the dotted lines on the graph, we were pleased to see this, actually are from the human safety trial. And the levels of blood acetyl-CoA in the humans corresponded nicely with the higher level of the compound that we measured in mice. We know that that dose effectively increases the CoA in the brain in the animals. So in summary, our animal studies showed that BBP 671 gets into the CSF and gets into the brain and elevates brain CoA levels. And in the next presentation, Dr. Dan Gretler is going to tell you about the results from the human phase 1 study and the next steps. So thank you very much to the organizers for the opportunity to tell you about our progress down this long road. And I'm very excited about the possibilities. Thank you very much.